going to do today is really focus on target discovery in AstraZeneca. We're using genome editing actually quite heavily from developing new tools, new methodology to use for developing preclinical models, understanding targets, discover and identify new target by CRISPR screens. So we're really heavily involved in genome editing. But what I want to focus today is really on how we use genome editing for target validation. And recently we have been really changing the way in which we identify and suggest target across disease area, but today I will focus on oncology. Previously, the way in which we tended to, the sources of new targets were really from collaboration, from literature, mining existing and public data set. But today we are seeing new targets coming from functional genomics, both internally and externally, from AI and machine learning, and also from data coming from patient and medicine and targeted drug in the clinic. Now, those targets are not necessarily well known, and there is sometimes not a wealth of information on them. And so we really need to validate those targets before we enter them in the portfolio. One of the main reasons is also that one of the main reasons for failure of clinical trials is actually a lack of efficacy. And that is very much based on the fact that target may not be very well validated. In these slides, it's really just to illustrate the failure that we often see in clinical trials with some of our drugs, and the reason why we really need to better validate our targets. Right. Mm-hmm. So this is really just, I'll skip quickly. This is just to, this slide's okay, thank you. So this is just to focus on the target validation pipeline in our drug development. And the example I will give today, can you go a slide back, please? The example I will give today, it's really focusing on how we try to establish the link between a gene and a disease, how we use genome editing to validate this link, and how we actually also use genome editing to understand resistance to target therapy, so how gene loss, gene variants can affect the response to target therapies. And the last example will be about how we try to predict what type of response we have in the clinic with our drug. So if you stay on this slide, here is actually an example of devalidation. So a specific gene supposed to be essential for small cell lung cancer, for non-small cell lung cancer, line with the KRAS and LKB1 mutation. And so how you can see here that we used cell lines, lung cell line with different genetic background to knock out this gene with two guides. And you can see at the protein level and the messenger level, we see reduction of expression of this gene. But on the bottom left, you can see that actually compared to control, we don't have any effect on the viability. So although this dependency was published using RNAi, by CRISPR knockout, we show that this gene is actually not essential in this genetic background, and therefore we didn't bring it in our portfolio as a new target. If you can go to next slide. And so this is really the basic type of work we do, but then we do also more specialized type of work. Can you go to next slide? Yeah, thanks. In which we try to understand actually patient data coming from resistance to our target therapies. And here you can see an example of a tyrosine kinase inhibitor, Tagrisso, that is used in lung cancer patients that have EGFR activating mutations. Can you go to next slide, please? And I have previously presented this data, but I would like to start from here to just illustrate what we do in those cases. We have a number of inhibitors for EGFR, but unfortunately, a patient can develop resistance, and in some cases, this resistance is actually due to a mutation in EGFR. And so novel inhibitors have been developed. Can you go to next slide, please? Novel inhibitors have been developed for these, and one of the things that we are trying to demonstrate is if, for example, those on-target mutations, and some of those you see illustrated there with a small molecule inhibitor that is an ATP competitor, if some of those mutations that we find from sequencing patient biopsies are really causing the resistance. Can you go to next slide, please? Next. And in the next slide here, I illustrate how we input activating mutation in cell line and then specific variant coming from patient data, and we show that this actually results in specific resistance to specific inhibitors. Can you go to next slide, please? We have also tried to accelerate this work by really trying to understand and predict other mutations. And here I'm illustrating a case in which we look at specific amino acid and we input every possible variance, every other amino acid. And like this, we show that we can actually predict clinical data, and we can understand which specific variant will actually cause resistance. Next slide, please. And we have brought this work even further, analyzing a number of clinical data like shown here, additional mutation causing resistance to tyrosine kinase inhibitor. Next slides, please. But also trying to do this type of work really across targets and across genes by mutating every single amino acid and creating every single possible variant and asking the question if these cause resistance to a specific drug. Next slide, please. And I just have a quick example to show, and these are proliferation curve. When, you know, in an assay like this, in each well, you have a specific amino acid and every possible variant for that amino acid in EGFR. And you can see that clearly, for example, in exon 20, we are able to identify only the mutation and the variants that have been actually found in the clinic. If you go to next slides, please. And so this allows us to really look across a gene. But also in the patient population, we found variants and gene loss across many genes. And so another setting in which we use genome editing is actually to test this loss of function by gene knockout in parallel, in which we look long-term assay like viability upon drug treatment, or even short-term looking at downstream signaling or apoptosis assay. If you can show me next slide, please. And here is again on resistance in EGFR-driven tumors, resistance to osimertinib. And in this next slide, you can now see that we have analyzed a number of genes, and some of these have really confirmed to confer resistance. And the difference between doing these and a genome-wide CRISPR knockout is that we're really validating here data coming from the clinic. If you can go to next slide, please. But the next and last bit that I really want to show you today is that we are also not only trying to look at variants in the resistance setting, but we have also tried to understand and to use genome editing to understand protein domains, their relevance in the signaling of the protein and their relevance in cancer. If you can go to next slide, please. And here I'm focusing on the RAS pathway, and specifically on the RAF genes that are central to these pathway signaling. They form homodimer and heterodimer, and they signaling from KRAS to downstream. Now, those genes, specifically BRAF, are commonly mutated in cancer, and on the right you can see that across BRAF gene, you have a number of mutations that are arising in different type of tumors, and these are across both phosphorylation site that are important for the regulation of this protein, but also the kinase domain and other conserved domain. Now, the relevance of some of those mutations, the functionality has been studied, but not all. And some of those domains have been studied in their importance for dimerization, both homo and heterodimerization, but not everything is known. And so we have used genome editing. If you can go to next slide, please. We have used genome editing to try to look at the functionality of those domain and mutation. And we have designed basically base editing guide across both BRAF and CRAF genes, as you can see there on the right. And these cover quite well the known domain, but also some less known regions. And the results I'm showing today are really focusing on a subset of those guides, because this is the first time we do this type of work. And therefore, we really focus on a subset of guides. And this was done in collaboration with Syntago, as they provided those guides for us. Next slide, please. And this, I think, it's really my final slide, in which I want just to kind of from the method point of view highlight that with base editing, we find, depending on the cell line, very high efficiency of being able to input mutation in a gene. But what we also find is that in the editing window, if there are more than one residue that can be mutated, we often find that. And also that we can really, due to the high efficiency of genome editing, really scan a gene and assay different downstream property. And for this, we have looked at homo and heterodomains, and we have also looked at downstream signaling, as I'm indicating here for CRAF. And we can look at the amount of protein by immunofluorescence. We can look at downstream signaling by immunofluorescence, and we confirm some of the known relevance of some amino acid in downstream signaling, but also we find novel relevance that we are gonna study further, and by comparing downstream signaling with the total amount of protein, we can also infer which are the amino acid that variants that actually result in less stable protein. If you can go to next slide, this is really the end of my talk, and I just want to briefly acknowledge all the colleagues in AstraZeneca, but also all the collaborators that have made a lot of this work possible. Thanks. I'm happy to reply to questions.
Thanks, Emanuela. I think we'll take one question. There's plenty in the chat if you have time to get to them later. Arun Bat wants to know, how do you control expression levels for different mutants so that it's an apples to apples comparison?
Okay, so we tend to do that by always checking both messenger RNA and protein level, we have noticed that genome editing can result in differences, modulation of protein, sometimes, especially with CRISPR-Cas, due to the fact that you may not only input a variant, but actually also inducing a knockout or an indels. So we tend to do that by messenger RNA and by protein levels, Western or immunofluorescence, as in the last case that I have shown. And we only draw conclusion by actually normalizing for protein levels.